All right, so here we are, and I'm going to share my screen to quiz statistics. All right. Again, I think it's worth noticing. One of the things that is useful about these is seeing kind of the range. Like if you're down here in the 40 or 50%, that doesn't mean you're not going to do well in the class, but it does mean that you need to be very conscious that you are not solid on stuff that you actually really need to be solid for, that everything else in this class is going to build on. If you are not doing well on this, you really need to take notice of that and do something active to get up to speed on the chemistry, because otherwise the rest of the class is just going to leave you in the dust, because everything keeps building on this. And also just to give you, so you can see we have a huge range from perfect scores down to less than half. So again, these are useful for you to know that, oh, this is a place you need to work on. So let's look at the actual things. So picture above illustrates various steps of the Krebs cycle and the mitochondria. We're going to be looking at all this very explicitly in the coming week. But we just have some molecule going to some other molecule, going to some other molecule, going to some other molecule. What do we call this when you have this chemical reaction that happens in multiple steps, one after another?
A pathway.
A metabolic pathway, yeah.
Yeah, the metabolic pathway. And we're going to see a bunch of these. And again, what makes one molecule transition to the next molecule in the pathway? What's necessary?
An enzyme?
An enzyme. There's always going to be a specific enzyme that takes you from one intermediate to the next intermediate to the next intermediate. Again, each, like citrate, isocitrate, alpha ketoglutarate, each of these are considered the intermediates, the molecules that you're making along the way. And then there's some enzyme that is going to catalyze the reaction from one intermediate to the next. And again, we'll look at the Krebs cycle as part of a much larger pathway when we look at cellular respiration in the cell. Describe the function of enzymes. What are their defining properties? Let's talk about the real essential ones and then other interesting ones, interesting qualities. What are the essential qualities of an enzyme?
It's typically a protein.
So let's put that as a secondary. They're typically made out of protein.
It's a catalyst.
It's a catalyst. It's an organic catalyst. And the definition of a catalyst is what?
Speed of reaction.
Speeds up reaction.
So one is it speeds up a reaction.
Speeds up the reaction. Mm-hmm.
And then what is the other essential quality of a catalyst?
Yeah,
It never gets used up.
It doesn't get used up. So it goes in, it makes this temporary enzyme substrate complex, but then it disengages and is ready to go again. That's another essential quality of a catalyst.
And they can travel to reactants or products without getting consumed.
Yeah, so it does not get consumed. It speeds up reactions. Enzymes, specifically the organic catalysts, are typically proteins. They don't have to be. There are RNA molecules that catalyze reactions. A guy, Tom Chuck, like back, this point, it's like probably 20 years ago, got a Nobel Prize for showing that RNA can act as enzymes as well. But all the enzymes we're going to look at in this class are proteins. And again, the other thing we talked about was this idea that the enzyme has this active site where the substrate binds, and that is an essential part of enzymes working. If something happens that changes the shape, denatures this protein so the active site no longer fits the substrate. That is going to affect the ability of the enzyme to do its job. So we'll talk a little more about proteins and denaturing proteins later today. Continuing. Which of the following would likely conduct electricity well? Wait, well, they should. So it included potassium.
Salt.
Yeah, so all the salts, sodium chloride, potassium chloride, the things that are ionically bonded, calcium chloride. Something like water. Does distilled water, conduct electricity well?
No.
No. It has a few charged things. We talked about there are some H pluses in there. That's why pH isn't, the pH is 7, but it's a very low amount. It's not gonna conduct electricity that well. Will oil conduct electricity well?
Definitely not.
Definitely not. There's no charged entities to move. And again, electricity is basically moving charge. If sodium, this one, okay, luckily most people got this. If sodium atom loses an electron, it is now a cation, a positive ion. Again, we're going to see Na plus all the time, and K plus, the potassium ions, and calcium plus plus, the calcium ions, Cl minus, which has gained an electron and is now an anion. And finally, this one, a lot of people got wrong. And this is going to be important to kind of get straight if you are confused about moles. 1 mole of an element weighs exactly the same as 1 mole of another element. Over a third of the people said true. What is a mole?
Number of atoms.
It's a particular number of atoms. It's Avogadro's number, 6.02 times 10 to the 23rd number of atoms or molecules. So if you have that many helium atoms, it floats. It weighs less than air. If you have that many lead atoms, it's really heavy, right? The weight of a mole of something really depends on what that particular element or compound is. So definitely, how do you actually figure out the weight of a mole of something?
Atomic mass.
Yeah, that atomic mass. You have to go to your periodic table and it tells you there underneath the letter how many grams per mole for that particular element. And if it's a compound, you have to just, it's the sum total of all of the atoms that make up that molecule. For sodium chloride, it's going to be like 58.5 grams per mole because that's the sum of the atomic mass of a sodium and a chlorine together. So that that should hopefully still be familiar to you from chemistry. And we still have at least a day's worth of chemistry review to go. So let's dig into that. So last class, we were talking about all sorts of different kinds of chemical reactions. Again, what were different types of chemical reactions we talked about?
Synthesis reactions.
Synthesis or anabolic, anabolism. What other ones?
Decomposition.
Decomposition or catabolism. Catabolic, when things break apart. What other kinds?
Redox, oxidation reaction.
Yeah, the reduction, oxidation, redox reactions, where you're transferring electrons from one thing to another. Usually there's energy being released, which is captured to do stuff. We talked about displacement reactions, where you just swap one part of one molecule onto another molecule. So that's one way to kind of think about them, like how are the atoms being rearranged? How are the bonds being rearranged? Another way you can think about reactions is energetically. We can talk about endergonic versus exergonic reactions. Endergonic means it needs energy to go. Like the reaction will not run unless you add energy to make it happen. Exergonic, this gives off energy. And these are typically coupled. Like a classic exergonic reaction that we've been talking about out the wazoo is, ATP breaks apart into ADP plus phosphate plus energy is released. This is the classic way of supplying energy to reactions in the body that are energy requiring. So often we will couple the breakdown of ATP to something else that needs energy to make it run. So this idea that you can use, again, exergonic reactions coupled to endergonic reactions to make things happen that wouldn't normally happen on their own without an input of energy. So we'll see the breakdown of ATP coupled into a variety of different processes as we go along. Now let's talk about specifying concentration. Again, most of the stuff we're talking about in this class is, atoms and molecules dissolved in water. Means we have to talk about, well, how concentrated is this stuff in water? During lab on Tuesday, we talked about the basic kinds we talked about true solutions, and we talked about colloids. We talked about suspensions, so I don't have to go over that again. But if I have, let's say I have sugar water, I have glucose in water, or I have salt water, I want to say well, how concentrated is it? What are different ways to specify the concentration of a solution?
Moles per liter.
So one of the major ways we do in a science class like this is moles per liter. Moles of solute per liter of solution. And what do we call that? What's the name for that?
Molarity.
Polarity.
Molarity. So if I say, and I usually use an underline, if I say 2 molar NaCl, that just means I have 2 moles of NaCl per liter of solution. Notice that this is a ratio, right? Concentration is not the actual amount of salt. It's how much salt per the amount of volume, right? The ocean, think about the ocean. If I take a cup of salt water, it's the same concentration as the entire ocean, right? And there's way less salt in my cup of ocean water than in the entire ocean, right? So the concentration is not the actual amount of stuff. It's the amount of stuff per specific unit volume. Again, the idea that the concentration of the entire ocean is the same as the concentration of a cup of ocean water, but there's way more salt if you look at the ocean in total as compared to in your little cup of ocean water, seawater, I guess. So just kind of remember that. So molarity we're going to use all the time. What's another way to specify concentration?
Osmolarity.
So there's osmolarity. This is one that you might not have seen in your chemistry class, but it's going to be very important for our class. So let's talk about osmolarity in more detail right now. It might be useful to do a quick aside to just refresh your memory about what osmosis is, since obviously from its name, this is going to be relevant when we're talking about osmosis. So let's actually do a quick little detour to reacquaint with osmosis. Not osmosis. Osmosis. You know, what is osmosis? What would be a definition of osmosis?
It's a molecule going through a solvent, or like what can pass through a semi-permeable membrane.
So you're so semi-permeable membrane is important. What particular molecule are we tracking?
Water, isn't it diffusion of water?
The diffusion of water across a semi-permeable membrane. You know, normally when we think of diffusion, we're thinking about some solute, like some sugar molecule diffusing or from some salt diffusing. But in osmosis, we have a semi-permeable membrane, which means, whoop, let me go back here. We have some membrane. And we have water on both sides. And water, we assume that water can pass freely across the membrane. That's why it's semi-permeable. But we assume also there are solutes. There are things that are dissolved. Maybe it's sugar molecules or salt or who knows what. So these things cannot cross the membrane. These are trapped on whatever side they're on. So semi-permeable membrane, semi-permeable, water can cross freely, but the solute, whatever solute is in there, cannot cross. So this is kind of our setup here. And in this case, so if there was no membrane there, what would happen? Here's my solute.
The salts would diffuse too.
Yeah, if there was no membrane, the solute would diffuse. The solute would go from where there's a high concentration of solute to where there's a low concentration of solute. It would go, the solute would go from side B to side A. That's what it wants to do. But it can't because there's a membrane there that blocks it, right? So this is kind of our setup here. If there was no membrane, the solute would diffuse from B to A because that's its diffusion gradient, but it's trapped. So instead, what's going to happen is we have this imbalance of water. So there's a few different ways to think about this. I'll talk first kind of more theoretically, and then I'll talk more how I do it more intuitively. If you think about actual diffusion concentration of water, which side here is more concentrated in water? Which one is more just pure water?
A.
A is pure water.
So this is, you could say this has a higher concentration of water than this side, because this side has water that also has lots of solute stuff in there. So which way is the water going to move? Which way? Water also has, is subject to diffusion. If we have a higher concentration of water here, lower concentration of water on side B, which way is the water going to go?
From A to B.
It's going to go from A to B. It goes from high to low concentration. That's just diffusion. So water is going to move from this place with less stuff dissolved to where there's more stuff dissolved. Intuitively, that's like wonky to me. That's like, because you don't think of concentration of water. You usually think of the concentration of the stuff dissolved in the water. So when I think of osmosis, I usually think about the saltier side kind of drawing in the water. You know, the water moves towards where it's more concentrated solute. If you were ever, I'm not proud of it, but when I was a kid, we'd get a kick out of sprinkling salt on a slug and then watching the poor little thing shrivel up. I don't know if people have ever done that. If that's basically, if you put salt on something, the salt kind of draws the water out of it. So when I think of osmosis, that's kind of more how I think about it intuitively. I think about kind of the salt kind of draws in the water. Like if you're trying to kind of get this intuition about how osmosis works, the water gets pulled into where there's more solute dissolved. So that's the basic idea of osmosis. And we need to think about this a lot. For a cell not to explode or shrivel up, we need to make sure that there's no net movement of water in or out of the cell. If I have some cell, you know, and water in general can move across the membrane. And if there is a net movement, more water moves in than moves out, this thing's going to inflate and explode. If we have a net movement of water out, this thing's going to shrivel up. So in order to make sure that there's no net movement of water, I'm going to have to make sure that there's a balanced number of particles, solute particles, inside versus outside. As long as there's a balanced concentration of solute molecules outside and a balanced concentration inside, then there's no net movement of water, and the cell is not going to inflate or deflate. So does that make sense? So we care about the concentration of solute particles, but we don't care what those are. And in fact, they're probably going to be different inside the cell and outside the cell. They are going to be. We're going to talk about differences in extracellular fluid and intracellular fluid. So all I care is about the total number of particles, no matter whether they're glucose molecules or sodium ions or whatever. Albumin, there's different proteins. So I am going to want to have some way to specify how many moles of particles are there in my solution. And I don't care what those particles are made out of. So that's what osmolarity is going to be. It's going to be our way to specify what is just the total moles of particles per liter without any concern about what they are. So now coming back to osmolarity, you've got a little more of a like kind of reason why we care about this idea. This is going to be moles of total particles per liter of solution. Again, as opposed to molarity, which was moles of the solute. So let's look at how this works. Let's say I had 2 molar glucose. Let's say I had 2 molar NaCl. And I want to know about the molarity. So I know the molarity. I know I have 2 moles of sugar per liter of solution, or 2 moles of NaCl per liters of solution. And then I want to think about, well, what does this mean in terms of osmolarity? The best way I know for doing this kind of thing is dimensional analysis. In dimensional analysis, you're basically kind of multiplying by identity. You keep multiplying by some conversion factor where the top and bottom are the same. So you're not actually changing the value because the top and bottom are the same. So let's see the way this works. I'll show you how this works. So if I think about case 1 here, by definition, what does 2 molar glucose mean? Oh, yeah. I usually abbreviate solution like SOL with a little N over. I saw the chat. So yeah, this is supposed to be solution. I realize it looks more like some little cartoon character. There we go. So what does it mean to be 2 molar glucose?
2 moles of glucose per 1 liter of solution.
So we have 2 moles of glucose per liter of solution. If I want to see it as osmolarity, I need to have moles of total particles per liter of solution. So the bottom is correct. The bottom of this thing, this liter here, that's what I want. If I can be more. But the top right now, I have moles of sugar, moles of glucose, but I need it to be moles of total particles. What happens? What's the conversion between moles of glucose and moles of particles when you put sugar into water? What happens to sugar molecules when you put them into water?
They dissolve, but they don't dissociate.
Exactly. Did everybody hear Morgan? It's going to dissolve, but they don't dissociate. They don't break apart. So every individual sugar molecule just becomes a particle, becomes a little thing floating around. So my conversion here is going to be 1 mole of particles is the same as 1 mole of glucose, right? Because sugar, the glucose does not dissociate. So if I have a mole of glucose, I have a mole of particles. A mole is just a number of them. If I had 10 glucoses, I'd have 10 particles. If I have 6 times 10 to the 23rd glucoses, I have 6 times 10 to the 23rd particles. In dimensional analysis, the reason I like dimensional analysis is you need to make sure that the units cancel. So it makes, if you're always trying to figure out, am I supposed to divide or multiply or whatever, as we'll see, it gets more complicated sometimes. You just make sure, all right, moles of glucose cancel. That leaves me with 2, if I multiply out on the top, it's going to be 2 moles of particles to liter of solution. So what does that mean in terms of osmolarity?
That will be 2 moles of glucose in 1 liter of solution.
This is going to just be what we call 2 osmolar. 2
2
osmolar,
osmolar.
because that means 2 moles of particles per liter. So does that make sense? So there it seems like, okay, why do we have osmolarity and molarity? But let's go look at what happens in the case of an electrolyte like sodium chloride. And let's start by definition again, 2 molar NaCl is 2 moles NaCl per liter of solution. And now we do this next step again. And again, we know on the bottom, we need to have moles NaCl. If I have 1 mole of NaCl, how many particles do I end up with?
2.
I'm going to end up with 2 moles of particles. Right? Because every individual NaCl actually dissociates into an independent chlorine and an independent sodium. So that means now that we have twice as many particles. So if I like multiply this out, 2 times 2, this is going to be 4 moles of particles per liter. So what is the osmolarity of this 2 molar NaCl solution?
Almost a particle per liter solution.
Say it again.
Almost a particle.
Yeah, or we'd say 4 osmolar. 4 osmolar, meaning 4 moles of particles per liter. So in the case of things that dissociate, osmolarity and molarity are very different. And this is going to be really important. In a few moments, I will illustrate why you definitely need to pay attention to this. You're going to kill somebody if you don't pay attention to this. So first, are there any questions about this basic idea about osmolarity and how it can be different from molarity? Okay. So then we can go back. Remember I said you need to make sure you have the same number of particles inside and outside to make sure that the cell doesn't inflate or deflate. So in general, the osmolarity of physiological fluids is 0.3 osmolar. So we often also just say 300 milliosmolar. Right, milli just means 1,000th. This is a number you definitely need to know. We're going to use it a bunch. Again, it's like knowing that body temperature is 97 degrees, or knowing that, I don't know, that water boils at 100 degrees, or I'm mixing and matching Fahrenheit and Celsius there, whoops. But this is like a core value that the body pH homeostasis is 7.4. You need to know, like if I have a cell, I'm going to assume that inside the cell is 0.3 osmolar. So if I want to make sure the cell does not inflate or deflate, I need to make sure the outside solution is also 0.3 osmolar. Isoosmotic, isotonic. We'll talk about all those terms a little later. But so it's going to be really important if we want the cells not to explode or shrivel up, we need to make sure that the fluid that they're sitting in is the same osmolarity as the inside, which is this 0.3 osmolar. I'm going to go through an exercise right now just to try to emphasize that this is not just some stupid esoteric stuff that you're going over, and that it's actually really relevant to physiology and to clinical practice. So we'll go do that in a few moments. Before I do that, I want to go over one last way of specifying concentration. So far we have talked about osmolarity. We've talked about molarity. What's another way to specify concentration?
Normal or normality?
Okay, so actually Lauren just said normality, which actually you should be aware of. We are not going to use this much. Normality is equivalents per liter. And equivalents, you should know what equivalents are. Equivalents, one equivalent, donates 1 mole of positive or negative charge. Normality is used often in specifying the concentration of acids and bases and stuff like that. It's also used, part of the reason I want you to know what equivalents are is if you're looking at the specification of dialyzing fluid for like dialysis, they'll talk about milliequivalents per liter of sodium in the solution. So if I have 1 mole of NaCl, how many equivalents would that be? It's a little confusing sometimes when you're first thinking about it if you haven't thought about it. How many moles of positive or negative charge get donated into the solution if you dissolve 1 mole of NaCl into the solution?
Two.
Two.
So you get 2 moles of total charges, but how much of either positive or negative?
1.
1. So again, when you talk about equivalence, it's one or the other. You don't put them together. So 1 mole of NaCl is 1 equivalent. What about 1 mole of calcium chloride? That's not what. What happens if you put 1 mole of calcium chloride into water? How much total positive or negative charge do you add in?
Well, it's 1 positive and 2 negative.
What's that?
It's 1 positive and 2 negative.
No. I mean, this thing wouldn't exist if it actually.
I'm sorry, 2 positive and 2 negative.
Right. Remember, because calcium plus and then 2 Cl minuses. So this would actually be 2 equivalents because it's adding either 2 moles of positive charge, or you could look at it as 2 moles of negative charge. So that's what equivalents are. Equivalents per liter is normality. Again, in this class, we're not really going to be using this more, but I think going into a kind of, oh, there's Munarich. So Munarich is now here. As you go into more of a clinical coursework, you are going to see things specified as milliequivalents per liter. And you're wondering, like, what the heck does that mean? That's what it means. It's like especially if you're talking about ions and stuff, it's a way to specify the concentration of charge per liter. But that isn't kind of an aside. That's not the main one I wanted to talk about. There's one more.
Will we be tested on normality?
You should know what equivalents are for sure.
Okay.
What other way do we have for specifying concentration?
Molality.
Say it again.
Molality.
So there is molality as well. We're not even going to go there. So that's like with weight and stuff. So let's not even go there. Let's just say percent solution is going to be our last one. Yeah, so Lauren, that is another one, but for our class, that's not going to be used at all. So percent solution. This is much more of a practical way, right? Because to do molarity and osmolarity and all this stuff, you got to get into this theory of moles, and moles are not something you can actually measure directly. You have to like weigh things and convert things, and so But if you're just like in a lab and you're trying to make a solution, you're going to weigh stuff out and mix it in the water. So percent solution is just, there's different kinds. We can do percent by weight, which is a classic way. So it's like grams of solute over grams of solution times 100. So it's just the ratio of the weight of the solute to the weight of the solution. And to make it a percent, you do times 100. You can also do percent by volume, which means, especially if you're trying to dilute something that's liquid, you can just say like how many mils of solute per mils of solution, but it's always just going to be this ratio. It's really easy to mix it up because how much does water weigh? So H2O. So if I have 1 mil of H2O, how much does 1 mil of H2O weigh? Anybody from chemistry? You should know this, I'd imagine.
I forget.
All right, so there's a few relationships around the metric units in water that you should know. Even what is a mil in terms of actual, like,
1 cubic centimeter. It weighs a gram.
Exactly. So if we have a box 1 centimeter cubed, that is a mil. And in fact, we say cc or cubic centimeters or mil. Those are all synonyms. Those are all ways to specify the exact same volume. If you get your little syringe, it might be, 1 cc or 1 mil or 1 cubic centimeter. Those are all the same thing. They're used.
A milliliter is 1 cubic centimeter?
Yeah, 1 milliliter is a cubic centimeter. These are all equivalent. These are all the same thing. And again,
But in medical charting, you don't write CC because you can confuse it for 00 and make an error.
right. But people often do say CCs and stuff. So like, or if you're listening to music, there was like a band that had CC in it. No. Yeah, so again, mil is the classic way to specify metric volume, but it is worth knowing that these are all the same thing. And 1 mil has a mass of 1 gram. So it's really a really nice conversion there. So if you want to know how much does 100 mils of water weigh, it weighs 100 grams. It's super simple. So you can get a good approximation of a percent solution just by taking, like if let's say I want 5% NaCl solution. All I need to do is get 5 grams of NaCl and dump it in, 100 mils of water, which is basically 100 grams. And, that's times 100, that's 5%. So in general, one of the beautiful things about percent solution is it's just really easy to make up. If you are in your like prep room and you just need to make 5% NaCl, just dump 5 grams of NaCl into 100 mils of water and you're done. There's no going to the periodic table and calculating grams per moles or blah, blah, blah. So a lot of times you're going to see solutions specified in percent solution because it is the more practical way to just make up a specified solution. And again, it's just grams of your solute over the, and again, in reality, if 100 mils, it's not going to weigh exactly because you're going to add this into this. And so the total weight of the solution, but it's close enough. It's close enough just to assume 100 mils of solution is about 100 grams here.
So if it was milliliters, it would be volume, and it would be, if it was grams on the bottom, it would be weight.
Yeah, and there's also, you can also do mass over volume. There's actually, when you look, if you look at this more depth, there's actually a whole bunch of different ways you can specify percent solution. So there's like 3 different ways. So what we're gonna do now is IV flush syringe and understand how all these fit together. And I have them in my office at school. But I have pictures. I'll show you a picture of one once we're done. So the idea here is, if I got, oh, here we go. I'm gonna have some syringe. And I'm going to take this thing and I am going to put it in someone's IV line and flush their lines. And I'm going to use salt water to flush the lines of the IV line, which means that whatever I am using to flush this line, this IV line, is going up into their veins, which means it's going to mix up into their plasma and all their blood cells are going to be bathed in it. So I need to make sure that whatever this saline, this salt water that I am injecting up into their veins, that it's going to be the right osmolarity, that it's not going to make their blood cells explode or shrivel up. So what is the starting point for this whole thing? If I want to make sure, I want to figure out what percent NaCl should I use to flush an IV line? What's my starting point for this whole thing going to be? If I want to make sure that the inside and the outside of the cell doesn't have a net movement of water in or out, what did we talk about?
0.3 osmolarity.
Mm-hmm. Remember this idea, if I want my cell to be happy, the inside and the outside have to have the same osmolarity. And we talked about the osmolarity of typical physiological fluids is 0.3 osmolar. So this is going to be my starting point for this whole calculation. I need to make sure whatever I'm injecting up into their veins is 0.3 osmolar. So that is going to be my starting, that is going to be kind of the starting point that I'm going to begin from. It has to be 0.3 osmolar or else it's going to explode or shrivel up this person's cells when you inject it into them. So let's start there. By definition, what does 0.3 osmolar mean?
0.3 moles of total particles per liter of solution.
Exactly, 0.3 moles particles per liter. So that is my starting point. Now I'm going to do this dimensional analysis, but we're going to get a little more complicated because we're going to have to do a number of conversions to get into this percent NaCl. So first thing, we have moles of particles. We need to convert this into sodium chloride, right? We have moles of particles, but we want it to be saline. So if I have moles of particles, but I want to convert it into moles of NaCl. What's the conversion factor there?
You put 1 mole particles on the bottom.
So I'm going to have moles of particles on the bottom.
And then moles NaCl on the top, but I don't know what it is, the number.
So
2
what
moles, 2 moles NaCl on the top and 1 mole particles on the bottom.
is that?
Or the opposite.
Right? Every mole of NaCl becomes 2 moles of particles, right? Because each individual NaCl breaks into 2 individual ions that are independent. So 1 mole of NaCl is 2 moles of particles, right? Yeah, stop me if any of this should make sense to you. The nice thing about dimensional analysis, it's like, are you supposed to multiply or divide by 2? We have to make sure that the units cancel. So I know that this 2 moles of particles has to be on the bottom because I need my NaCl on top and the particles need to cancel. Next, percent solution NaCl, I need to be in grams of NaCl, not moles of NaCl. So I need to convert this into grams of NaCl. What's the conversion between grams and moles of NaCl?
Molar mass.
Say it again.
The molar mass.
Exactly. And does anybody remember what that is?
Well, it'd be the molar mass of sodium and chlorine, but I don't have those numbers.
It's basically 58.5 grams of NaCl for every mole of NaCl. Again, in that you just get straight off the periodic table, right? It's sodium plus chlorine's atomic mass added together. So now moles of NaCl cancel out. And again, with this dimensional analysis, if you ever get confused about what number's on top, what's on bottom, make sure your units are lined up appropriately to cancel. So right now we have grams of NaCl per liter. So let's just multiply this out. If we go here, actually we can have, somebody have a calculator right on them right now? Someone can do it for me. I see Marissa reaching there.
So it's 8.8.
8.8. And on the top, we're going to have grams of NaCl. And this is per liter. All right. And then we can say, okay, so we are almost there. We need percent NaCl. So this is going to be 8.8 grams of NaCl. How much does 1 liter of water weigh?
A 100?
1 liter is how many mils?
1,000.
Oh, it's 1,000.
1,000 grams.
It's going to be 1,000 mils per liter. And we know that 1 gram of water per each mil, right? So it's going to be 1,000 grams. Right, that should be pretty intuitive. If a mil is a gram, a milliliter is a gram, then a liter, which is 1,000 milliliters, should be 1,000 grams. And then for percent solution, we just have to do this times 100. And this is going to be 0.9% NaCl. So does this make sense? I should ask, is there anybody that has questions about what I've just done here?
If you left it in liters, would it be percent by volume?
Yeah.
Okay, but you wanted percent by weight.
Yes.
Okay.
And so if I go here, in fact, here we go. I'm going to go view image here. I'm going to share my screen here. You know, normally I would just throw these out at you, but here, this is a saline flush syringe. And what does it say on there?
0.9%.
0.9% NaCl. So that's not just random. It's not, they didn't just like, oh, let's like, yeah, that sounds good. How about 0.9% NaCl to flush the IV line? This is this very intentional concentration that is balanced with your osmolarity of your inside your cells, so your cells don't explode or deflate. And you can't come up with that 0.9% unless you actually think about it in terms of osmolarity, and then osmolarity related to molarity, and molarity related to actual grams of NaCl. So just trying to kind of motivate like why you should care about osmolarity. Like, you know, if you didn't think, if you're not actually putting in the right concentration of saline up into this person's veins, you're going to damage them. And so that 0.9% is exactly what they need. So the cells are bathed in 0.3 osmolar solution so they don't inflate or deflate. So are there any questions about that? Okay. So I'm seeing more chats. Let me, what have people said? Oh, okay. So stop sharing. All right, so that is stuff about specifying concentration. Next week, we're going to be talking way more about the idea of osmolarity and osmosis and hyper and iso and hypotonic and hyper and iso-osmotic and all of that. But for right now, this is kind of your first kind of introduction to osmolarity and realizing, you know, again, we're going to have 2 entire labs that are just going to look at osmosis and diffusion because they're so critical to everything we're doing in this class. All right, so now we can get to the basic types of biomolecules. This will be kind of the last part of our chem review, is making sure you understand the basics of proteins and carbohydrates and lipids and nucleic acids and things like that, and what are their basic properties and all of that. So again, this, if you were in Chem 110, you've seen this before. If you were in Chem like 114, you might not have gone into these, but you'll have a good background as we go through this to just understand it pretty easy. In fact, I usually find people who've taken Chem 114 have a much easier time with the chemistry because they went deeper into the fundamentals. So they actually understand it better. So as you add more things in, they can just grasp it quickly. Whereas Chem 110, they try to do general chemistry, organic chemistry, biochemistry, and shove it all into one semester, which is absolutely impossible. So you get tons of stuff coming at you and not a whole lot of it sticks as well. So I do have, I do empathize if you're coming out of like Chem 110 and you're like feeling like I'm still getting some of the core concepts here. So what are there's 4 basic kinds of biomolecules that we are going to be working with throughout the semester. What are the 4 basic organic molecules we deal with, classes of organic molecules?
Carbohydrates.
So carbohydrates.
Proteins.
Proteins.
Lipids.
Nucleic acids.
Lipids.
Nucleotides.
Nucleic acids, or yeah. So we're going to look at each of these. Let me look at the time. Let's start with, we can start with proteins. No, let's start with carbohydrates. We'll start with carbohydrates, take a break, and then go and continue on with some of these other ones. So let's start with carbohydrates. So carbohydrates. Carbohydrates are always going to have carbons, hydrogens, and oxygens, and they're always going to be in the same ratio. The ratio is always going to be 1 to 2 to 1. You know, as opposed when we get to lipids, it's going to have the same atoms, but very different, very different ratios. There's going to be way more carbons and hydrogens compared to oxygens. So, in fact, they get their name because when people first knew about the ratio, they said, oh, it's carbons and H2s and O's, which is kind of like water. Carbon and hydro. Hydro is water, but they're not actually carbons and waters. But they have the ratio of carbons and H's and O's as if it's like carbons and waters. So if you're wondering, what does that name even mean? It just means like carbon water or whatever. There are 3 main subclasses in the carbohydrates. Monosaccharides, also we call simple sugars. This includes glucose, right? And actually C6H12O6, depending on how you hook them up, this could be glucose. This could also actually be fructose. It could be galactose. There's a variety of ways to connect those in to make different versions of sugar. They tend to zip around in a ring where 5 of the carbons are in the ring, and then there's an oxygen, another carbon is off on one of the coming off. So when I draw glucoses, I'll usually draw little rings. This would be an example of a hexose sugar because it has 6 carbons. There's a lot of pentose sugars that are important. Right, it's still the same ratio, 1 to 2 to 1, but now it's 5, 10, 5 instead of 6, 12, 6. What are some of the important pentose sugars that we see in our class?
Deoxyribose.
Yeah, exactly. This would be like ribose and deoxyribose. So those, ribonucleic acid and deoxyribonucleic acid, DNA, that's part of the backbone of those molecules are these pentose sugars. So these are examples of monosaccharides. If you attach 2 monosaccharides together, you get disaccharides. So we have basically 2 sugars together. And depending which ones you connect together, you get different ones. If you have a glucose plus a glucose, you get maltose. If you have a glucose plus a fructose, you get sucrose. Sucrose is particularly important because it's one of the main transport sugars in plants. And we get a lot of our energy from eating plants. So we get sucrose from a plant. And then if you want to turn that into glucose, you've got to break that bond between the glucose and the fructose. Then you free up a glucose, and then you can transform the fructose into a glucose. You can have glucose plus galactose. And that becomes lactose, milk sugar. So there's a variety of different disaccharides. This is as good a time as any to talk about the terminology like hydrolysis, because we often talk about hydrolysis kind of when we talk about kind of breaking things apart. So for instance, if you have, so here's some sugar, and I'm just, to make my life easier, assume like the main thing has a ring and there's a hydroxyl off of it. Here's some other sugar, and it's got a hydroxyl hanging off of it. If you want to connect them, you're going to have these, they connect up where these two hydroxyl groups are hanging off of these sugar molecules. And you'll end up having just a single oxygen in between them, which means two H's and an O went away.
I don't need help with this.
So sometimes they call this dehydration synthesis even.
I don't need help with this.
Because you're putting them together, and in the process, you're losing the water.
Helping people.
What are some ways can help God's creation?
If you want to break this apart, you have to add a water in. So if I want to go the other way, I can add in an H2O to recreate the two individual monosaccharides. So when you see the word hydrolysis, hydro, water, lysis, cutting apart, it gets its name because you have to add a water in to actually accomplish this breaking apart into two individual molecules here. So you'll often see the word hydrolysis used when you're talking about kind of breaking things apart. It just means water cutting or whatever. Okay. All right, so monosaccharides, we'll see a bunch, particularly glucose will be our, our constant companion through the semester. Disaccharides, particularly important, sucrose, coming, that's, CNH, pure cane sugar from Hawaii, grown in the sun. That's sucrose. You get it from sugar cane or from beets and things like that commercially. Finally, there are polysaccharides, when you attach a whole bunch of monosaccharides together in big long chains. Polysaccharides. So if I have a whole bunch of glucoses all connected in a big long chain, one thing we get, like in plants particularly, you get cellulose. Oh no, actually let's start with starch. So starch. This is important for us because this is the main storage carbohydrate for a plant. And we get a lot of our energy from eating plants, like I said. So like, there's a potato plant, and this potato plant is like, I better put away some energy for the future here, and I'm gonna make lots of starch and store it in my tubers. And then like, the human comes along and it's like, oh, yum, I'm gonna pull this thing up and kill the plant and eat this and get the energy stored in the starch that this plant put away. Same thing like in wheat. The wheat's got carbohydrate in there, And it's kind of there for the little new seed to grow into a new plant. But instead we harvest it and we grind it up and we make pasta out of it. So starch is just a carbohydrate made out of lots of glucoses all connected together. And then if we break the bonds in between these, we're freeing up glucose and we got the fuel for our cells. In our body, we have a version of this called glycogen. Glycogen is very similar to starch, but instead of kind of more of a linear, it's more branching. So each of the glucoses but it's still like really easy to release them. Like these are going to be stored in your liver cells to keep your blood sugar balanced. This is in little granules in your fast twitch muscles so you can get energy quick through glycolysis. Sometimes people call this animal starch. So it's the same basic idea as plant starches, except it's more branching like this. The one other polysaccharide that you should know is cellulose. This is in plants, it's usually more structural. And it's also glucoses connected together, but it's in more of a, maybe I should just, the bonds. So glucoses, bonds between the glucoses can't be broken by human digestion. So in starch, whoops. In starch, you eat a potato or something, your body can break apart all these glucoses and free them up and you get energy. However, in cellulose, you eat cellulose and your body cannot break it down. So it goes through your digestive tract and it comes out in your poop. This is often what we call the fiber or the roughage in your diet. There are bacteria in your gut that can break it down to some degree. It's, again, when you kind of get like gas from eating things, it's, bacteria breaking down this. Other animals that have to get nutrition from this have more complicated digestive tracts than we do. If you are a cow, for instance, and most of your energy is coming from eating grass, then they chew the grass and they swallow it, but it goes into a different stomach. They've got like more than one stomach. They have one of their stomachs filled with bacteria that actually break down the cellulose in the grass and actually free up that sugar. And then they regurgitate, chew their cud, and then they swallow it into a different stomach where they actually are going to be able to kind of absorb the sugars. So it is possible to get energy from cellulose, but it takes more complicated digestive tracts. Again, the ruminants, the animals that are the ruminants, they have a symbiotic relationship with bacteria in their digestive tract. And the bacteria do have enzymes that can break apart cellulose to free up the energy. But for us, it's just the roughage or the fiber in your diet. So any comments or questions about all of this? Maria, or are you just waving? Oh. All right, so let's take a break. We are going to return at 10:10. If you do not have your pre-lab to your jumping lab done, please do that. And so we're going to do the jumping lab. We're also going to talk a little more about the chemistry and also talk more about numbers, about precision and accuracy and things like that. So let's take a break, shake it off, and we'll regroup in a half an hour.
Professor, can I ask you a question while people are on break?
I'm sure.
Okay, I'm a little confused about hydrolysis, and just want to make sure that I'm understanding it correctly. So the disaccharides, so two monosaccharides together are like each one has a hydroxide that's attached to it.
Hydroxyl? Yeah.
For some reason in my chemistry class, I feel like we call it hydroxide, the OH minus.
Just a free group, an OH
Yeah.
functional group. I think you just call hydroxyl.
Oh, okay. Hydroxyl. I don't know why we. So then they combine together, and then to make them two separate again, you have to add in a H2O. Is that the idea? Or like, can you just go through that one more time?
Yeah.
Okay.
So this idea that if you want to recreate the two individual sugars, they're bonded here, this little corner of the ring where they're connected with just a single oxygen. At the end of this process, you want to have an OH on the vertex of each of these, which means you only have a single O here. You need to add another O and two H's in order to recreate these two individual monosaccharides.
Right.
So that's coming in through a water.
Okay. Because before when they got it. Okay. And so hydrolysis is coming in to break apart into two molecules.
Correct.
I get that. Another question. I have never heard of normality before, and so the concept, like the equivalence, I'm a little confused about you saying that the donating the positive or negative charge.
So just anything that a mole of positive charge or negative charge is going to be one equivalent.
Okay.
So equivalent just is, it's a mole, like, so sodium chloride would be one equivalent because it's, or 1 mole, I should say, 1 mole of sodium chloride would be one equivalent because it's donating 1 mole of charge, whether positive or negative. Right? 1 mole of calcium chloride is 2 equivalents because it donates +2 or -2.
Okay. And so we don't need to go into like who it's donating to. It's just the concept of like its potential for donating and when it's.
Or it's adding it to the solution.
Okay.
Yeah, it's just adding those charges into the solution.
Okay.
Because this is a concentration, way to specify concentration. So
okay.
Charge floating around in a bit more regarding chemistry. So we just reviewed, we talked about carbohydrates. Another major player in this class will be proteins. How would you describe a protein? What are the essential qualities that make something a protein?
The chain of amino acids.
And it has nitrogen.
So say that one more time.
It has nitrogen, a difference than the other molecules.
So it has nitrogen as well as other things. There's something probably more. Yeah, that's part. Yeah. So in fact, what do we call like a nitrogen that has a couple of H's on it? What's that functional group?
Amino group.
Amino group. So the building block of all of our proteins are going to be amino acids. So proteins, the building block of proteins are going to be amino acids. So to understand proteins, we should probably just make sure everybody is on board with what's an amino acid. So amino acid, got an amino group. And the acid is a carboxyl. And then there's some side chain here. So this is an amino acid. They all have this same basic structure. The acid, again, the carboxyl, the amino group, and then the side chain. Every amino acid has a different side chain, and the different side chains give the different amino acids different kinds of chemical properties. Makes them want to make different kinds of bonds. Some side chains create amino acids that like to hydrogen bond. Like some have side chains that make them want to receive electrons or give up electrons to become the positive or negative sides of an ionic bond. Some side chains are nonpolar and make it so this amino acid doesn't tend to participate in these types of bonds. So that's going to be really important when we start talking about this idea of binding. Like, what does it mean for a enzyme to bind its substrate? Or what do we mean when we say a receptor is binding its ligand, its neurotransmitter or hormone? All of the receptors in your cells are proteins. We're going to be seeing how different amino acids hooked up in different ways present different side chains that like to make different kinds of bonds. So that's important. In a human, there are 20 amino acids to make up all the different proteins that are in your body. There are other kinds of amino acids as well out there, but in terms of the ones that make up all the proteins in your body, there's 20 of them. And we'll talk more about them. Then obviously we need to start hooking them together, and you basically hook them up end to end using peptide bonds. So what we call the primary structure of a protein is just which amino acid is hooked up to which amino acid. We start hooking them up in big chains. Dot, 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 dot. And again, the bond, you don't need to know the details. It's basic. These are called peptide bonds. What determines, how does the cell know which amino acid to hook up to what other amino acid to make the protein it's trying to make?
Messenger RNA?
Yeah, exactly. That is the messenger RNA, which you transcribed off of the DNA. Remember, it's like there's transcription, then you have to do the editing, cut out the introns and all that stuff. And then you do the translation at the ribosome, and basically the transfer RNA brings each amino acid one by one to the appropriate codon. So that's hooking these up. Depending how many you hook up, if it's just a few, like 10 to 100 or so, we usually call it a peptide rather than a protein. Peptide. If it's greater than 100, then we start calling it a protein. Some of these proteins are huge, like tens of thousands of amino acids all connected together. But some of these smaller peptides are super important. They are going to be playing important signaling roles, like a lot of the, insulin. Insulin, everyone's heard of insulin. Insulin is just 37 amino acids connected together. It's a peptide. A lot of the basic signaling molecules in your nervous system, the ones involved in the pain pathways, like the naturally occurring ligand that binds your opioid receptors, are little peptides. So some of these peptides are going to be super important. The larger ones are going to be playing different roles. They'll be playing, what kind of roles are proteins playing in your body?
Enzymes.
They're going to be enzymes for sure. What other roles are they going to play in the body?
Transportation.
They're going to be transport. These are going to make the different channels that help move things in and out of cells.
They're going to be structural.
It's going to be a lot of structural, just your connective tissue, all the little fibers that hold things together. There's going to be other proteins that are going to be the contractile proteins of your muscles. So some of them are going to be more functional. There's going to be other receptors on your cells that are proteins. So they're doing both structural and functional. So structure and function, just kind of, they're playing lots of roles. Some of them are more fibrous, some of them are more globular. So primary structure is just the order of amino acids, which amino acid is hooked up in which order. But then that's just the very beginning. The next step is these things start folding. And it is important to understand the basic folding. So let's start with the secondary structure. Secondary structure, there are two main types of secondary structure. One is called the alpha helix. The other is called beta pleated sheets. Whoops. In the alpha helix, let me just basically every third amino acid hydrogen bonds to the one below it in the spiral. So you just end up getting these big spirals. That's why they're called helix. So 1, 2, 3. Yeah, so every 2, 3, every third one just binds to the one below it, and it holds in these stable helices. Beta pleated sheets, instead of looking like this, they hold more like this. So they kind of go face to face in these big sheets where they hold on to each other with hydrogen bonds. Remember, hydrogen bonds, we talked about being between two different molecules, like holding two water molecules close to each other. But you can also have hydrogen bonds between two parts of the same large molecule. So here we have hydrogen bonding between different parts of the exact same molecule. Same way DNA, hydrogen bond is holding the A to the T or the C to the G. So hydrogen bonds can be between different parts of the same large molecule. Or as we've seen with water, they can hold separate molecules together. So this is the secondary structure, these alpha helices and these beta pleated sheets. But wait, there's more. Then we get to the tertiary structure. Tertiary structure, you get much more complicated folding. Like you get some areas that are beta pleated sheets, some areas that are helices. So this is this much more complicated folding. Folding. Here you have hydrophobic, hydrophilic interactions. Right, some parts of this thing, like, there's water around here. Some of these things want to get away from the water because they're hydrophobic. Some of them want to be near the water because they're hydrophilic. So that's going to influence whether, what parts of it fold outward or fold inward. There's little things called disulfide bonds. Different areas where different parts bridge across to help stabilize this more complicated shape. So there's different things that influence the overall structure. This many proteins are functional at this level. Like a lot of the enzymes that you've met, are just a big globby thing like this. Sometimes they actually require some helper proteins and stuff to actually fold properly. Later on, we're going to talk about like prions and things like that, which are, cause horrible neurological diseases, and they're coming from proteins that are folding improperly. So tertiary structure is this more complicated folding. Some proteins require more than one subunit connected together to make the functional protein. So this is optional, but it's going to be some of the important proteins that we see. So this is optional. You can have this tertiary structure. So this is two or more, quaternary, the 4th degree structure. 2 or more tertiary structures to make, kind of subunits to make the functional protein. For instance, when we look at hemoglobin, the classic thing that's moving the oxygen around in your blood, it's 4 globin subunits to make a functional hemoglobin. When we look at myosin, there's going to be the heavy chains and the light chains to make a functional myosin. When we look at antibodies, there's going to be heavy chains and light chains to get together to make a functional antibody. So this 4th level quaternary structure is when you have 2 or more subunits that you need to assemble together to make the functional protein. So that is the basic idea of proteins. And let me, hold on. Okay, so I'm gonna share. Slideshow. Right. So again, showing this is a basic amino acid. It's got the amino group, it's got the carboxyl group, and it's got a side chain coming off of it. And again, there's many different kinds of side chains to give them different properties. You can start connecting the carboxyl and amino sides together with a peptide bond to start making big chains of amino acids. Now you're starting to, this is supposed to be like, lysine and proline and tyrosine and phenylalanine and glycine. These are all arginine. These are all kind of the classic amino acids that you find in your body. So again, what would we call this structure where you're starting to just string them along?
Primary.
That's the primary structure, just which one is connected to which. But this is showing how you start getting the secondary structure, either these alpha helices where every 3rd amino acid hydrogen bonds to its second, to the one below it, the beta pleated sheets where you have one side kind of just getting attracted to another long chain across this way with hydrogen bonds. So you can have, and then tertiary structure is where you have all of that happening and you make a big complicated structure that is, the functional protein that has a very specific shape and very specific chemical properties depending on which kind of amino acids are showing here, right? Some amino acids are going to be hydrophobic or hydrophilic or want to make hydrogen bonds or make ionic bonds. So this thing has a very specific shape, and it also has very specific chemical properties, like what kind of bonds the different amino acids that make this thing up want to make at the different sections of it. Again, quaternary structure is when you have two or more, in this case, hemoglobin, you have 4 of these subunits connected together to make the functional hemoglobin. This is kind of just showing some of the stuff in the tertiary structure. You can have hydrogen bonding, you can have these disulfide bridges, salt bridges. Salt bridge just, you know, like I said, some amino acids like to give up electrons, some like to take in electrons. That can happen across the exact same huge, again, one of these proteins can be tens of thousands of amino acids long. So there's lots of opportunities for one section of this thing to grab on and hold on to another section to create its specific shape. Oh, no, so let's leave this. Stop share. So any questions about the basic, what is the structure of a protein? So then the last thing we should talk about is denaturing. Denature, I think just denaturation. I don't know, you just, you know, what does it mean to denature a protein?
Break apart.
Break it down.
Good.
So you either break it apart or make it lose its shape. So remember I talked about all that folding to get that very specific structure. If something happens that messes with that shape, we say that the protein's denatured. What kind of things can denature a protein?
Heat.
Heat.
pH.
Changes in pH. Again, if you think about that, you know, hydrophobic, hydrophilic interactions, if you have higher or lower pH, there's a lot more positive or less positive charge around you if there's not as many hydrogens or more hydrogens. And that is going to either squeeze things in or fold things out. So pH can denature, can mess with things. Heat, because things are now moving around. You have all these chains of amino acids and they're wiggling more. The more heat, the more energy. And all of a sudden they're getting all tangled with each other. And now they're no longer in their nice folded thing. They're now just a big tangled up mess of amino acids. What else can denature things? Denature proteins, I should say.
Radiation.
Yeah, that actually can. That kind of just rips all sorts of stuff apart. I'm going to say you can actually mechanically do it as well. Examples just in your life experience. If you think of egg whites are albumin. It's just a protein. We're going to meet albumin later as well. So you have albumin, you have these proteins, and they are making this gloopy, gooey egg white. But if you heat it up, then all of those things get all tangled up into one big white solid block. You know, that's because you've denatured all those proteins and they're all now kind of inextricably tangled up together because the heat. That's when you kind of cook your egg. You can mechanically denature the egg whites by whipping them, and that's how you get meringue. You're basically physically just making these things go whoa, whoa, whoa until they're all tangled up and making stiff peaks that you can then spread on top of your lemon pie or whatever. pH, classic is ceviche, right? You take a fish, if you want to cook fish protein, you could put it in the oven, or you could just put it in with some lime juice and let it sit in the lime juice for a while, and it will also cook, cook. It'll end up being denatured. The flesh will go from translucent to more of the flaky white and all that. So particularly because, oh, hold on, I just, cannot, okay, I just, Oh no. Video. Something fell here and now I'm having trouble getting my video started again. Oh, there we go. I'm back. So proteins make up so many important things in your body, your enzymes, for instance. And again, an enzyme, if you have some enzyme who is a protein, and then you have that active site, that binds the substrate, that shape is super important. Right? If anything happens, if there is heat or changes in pH or something, that can totally denature this enzyme, change the shape of the active site, and now this thing is no longer going to be able to function. Right? So that's going to be a real important thing going on in your body, is making sure we maintain an environment that is not going to denature the proteins in your body, not going to deactivate the enzymes that are running all the chemical reactions you need to stay alive. So yeah, it's important to understand this idea, denaturing a protein, again, which just is another word for making it lose its shape, is going to affect the ability of, for instance, of an enzyme to bind its substrate. So then it can no longer do its job. Any comments, questions about that?